Let's jump right into this. Today, we're unpacking a groundbreaking 2026 benchmarking study from Nature Microbiology. And honestly, it's sending shockwaves through the biology world. If you're already following the latest in microbiome wrapping, you know exactly what's at stake here. Today, in this explainer, we're looking closely at the tools we actually use to map these microscopic universes. And we're going to see exactly where this massive promise crashes into some very messy reality. Okay, let's dive into this. Imagine taking 70 completely different, incredibly complex microscopic organisms. You toss them all into a biological blender, hit puree, and then you have to figure out exactly what went in, right? That is literally the everyday challenge of microbiome research. You're handed this massive, tangled soup of DNA, and you've got to sort it out. Historically, we've relied on two main tools to solve this puzzle. Think of our blended microbial DNA like a massive library that someone just ran through a paper shredder. Deep shotgun sequencing? That's incredibly expensive but highly detailed. It's like painstakingly reading every single shredded page. Then, on the flip side, you have 16S sequencing. Super cheap but incredibly low res. You're basically just reading the titles printed on the spines of the books, totally missing the actual story inside.
Section 1. The Microbiome Gold Rush. Researchers everywhere were desperately looking for a middle ground here. They needed something that gave them the cost-effectiveness of 16S, but the deep, functional insights of shotgun sequencing. Enter our main subject for today, shallow metagenomics. This was championed as a cheap miracle tool. The definition is pretty simple. You sequence your sample at 1 gigabase or less, which is roughly 3 million reads. Going back to our shredded library analogy, this is like skimming just a few handfuls of shredded pages and hoping, crossing your fingers, that it's enough to perfectly understand the entire library. Moving to Section 2, the ultimate benchmark test. But does skimming actually work? The researchers behind our source material decided to put this massive industry hype to a rigorous stress test by building basically the ultimate benchmark. Now, what's really interesting about this slide is that number right there, 50.0 gigabases. To discover exactly what shallow sequencing was missing, they couldn't just sequence shallowly. They sequenced all the way up to a massive 50 gigabases just to establish a flawless, undeniable baseline. To pull this off, they created 13 highly complex mock communities. You can see the core groups here. They mixed exactly 70 strains or 24 strains and distributed them either perfectly evenly or staggered, meaning some strains were super dominant while others were barely a trace. Because they knew exactly what went into the blender, they could definitively score the software tools on how well they reconstructed reality. Section 3, Success with a Map. The study actually brought some fantastic news right out of the gate. Shallow sequencing works beautifully. But, and this is a massive but, it requires a map. Having a master copy makes all the difference. In bioinformatics, we call these reference genomes. If you already have an unshredded master copy of every book, comparing your tiny handful of shredded pages to that known source works perfectly. In fact, the researchers found that accurate reference-based strain taxonomy was easily achieved with as little as 0.5 to 1.0 gigabases of data. That's practically nothing. So, if you know what you're looking for, and you have a solid database, shallow metagenomics is an absolutely crucial, highly cost-effective tool to map out who exactly is in the microbiome. Section 4, the Frankenstein genome problem. So the crucial point is, what happens when you don't have a map? This is where the promise versus reality arc of this study really hits its climax. In the real world, researchers are constantly discovering new bacteria that don't have reference genomes. So they build MAGs, or metagenome assembled genomes. This means reconstructing genomes completely from scratch. Imagine trying to tape those shredded pages back together to write a cohesive book you've never actually read. The results here are, frankly, shocking. When the researchers looked at what the software confidently labeled as high-quality MAGs, which, by the way, is supposed to be the gold standard, nearly half of them, over 45%, were actually chimeric mix-ups. They were literal Frankenstein creations, blending sequences from completely different bacterial strains into one totally fake profile. Let me share a specific, horrifying anecdote directly from the study's data. We'll call it the 12-genome monster.
The researchers found one single, high-quality mag that was actually composed of DNA fragments stitched together from 12 completely different reference genomes. The software proudly presented this as one distinct organism, but it was just a total biological fiction. No way that exists in nature. Now, you might be thinking, well, just sequence deeper. Throw more data at it. But look at this quote from the authors. Deeper sequencing does not guarantee more complete mags and does not eliminate chimerism. Throwing more money at the problem and sequencing up to 10 or 50 gigabases, it didn't fix the core issue at all. It often just created more split, fragmented Frankenstein genomes rather than actually solving the puzzle. Section 5. Messy labs and missing functions. Moving beyond just identifying the bacteria, we run into the next major limitation of shallow skimming, finding out what these organisms actually do. Shallow skimming completely misses the deeper functional reality. If you want basic biological pathways, you're going to need 2 to 5 gigabases. But if you want full proteome coverage, meaning you actually want to see all the functional proteins the bacteria could produce, you need well over 10 gigabases. Shallow just doesn't cut it for those deep functional insights. And this brilliantly illustrates where things go wrong in the physical pipeline. Before you even touch a computer, you're in a wet lab. The study found that shallow data is highly vulnerable to physical corruption. The raw volume of input DNA, the number of PCR amplification cycles you run, and interference from background host DNA, like from the mouse guts they used in this study, all of these heavily distort the final data when you're only sequencing shallowly. By strictly comparing 2 different sequencing facilities, the authors provided some very practical, actionable takeaways to mitigate these wet lab errors. To get robust data, you should use a higher template DNA input, like 100 nanograms. You absolutely need to minimize PCR cycles to reduce amplification bias. And if there's high host background DNA in the mix, you simply have to increase your sequencing depth to overpower that noise. Section 6. The final verdict. Let's move to and see how this builds to our conclusion. What is the ultimate reality check for microbiome researchers today, based on this rigorous data? Well, here's your core takeaway. Shallow metagenomics is undeniably powerful and cost-effective, but only if you have a map. If you're building from scratch, it artificially inflates bacterial diversity. Today's massive MAG databases, they must be handled with extreme caution, because we now know for a fact they contain a significant number of chimeric mix-ups. Which leaves us with a rather profound question to ponder as we wrap up this explainer. If our standard gold-tier tools are routinely building Frankenstein genomes that don't even exist in nature, how much of our currently mapped microbial world is actually just an illusion? Think about that next time you read a headline about a massive new microbiome discovery. Thanks for joining me on this one, and keep questioning the data.